Adaptation, Acclimation, and Refugia, How Mosses Survive and Thrive in the Desert. I'm going to give a bit of background on mosses in general, mosses in the desert, and then get into some of my recent research in this area. To begin with, it's important to talk about mosses because they're not like other plants. Of course, they have a lot in common with land plants, but the ways that they're different have really important implications, especially for desert mosses. So the life cycle, it's the same alternation of generations that all land plants go through, but the different part is that the dominant stage of the life cycle is the haploid gametophyte in mosses, and they reproduce with swimming sperm and egg. So sperm has to leave a male gametophyte in species that are dioicous, separate male and female, and find an egg on a female gametophyte. Fertilization occurs, sporophyte is formed, and then dispersal occurs via spores, and the cycle starts over again. So on this gametophyte stage, sex is determined by a single chromosome, either U or V. And also, asexual reproduction is really common in mosses. There are different ways it can happen. Very common is fragmentation. So just breaking off a bit of the gametophyte can create a new gametophyte. So it's just a type of cloning. Some species make specialized asexual propagules and can clone that way. And some will self for those that are monoicous and make both sperm and egg. And that's really powerful when they do, because unlike tracheophytes, when a moss selfs with sperm and egg, all of the spores are identical to the parent gametophyte. So it's a really effective form of cloning. A really big one, though, is that water relations are very different in mosses than in other plants. Mosses are poikilohydric, which is a fancy word that means the cellular water content equilibrates to the external water content. So if it's wet outside, the moss is wet inside, and if it's dry outside, the moss is dry. This is as opposed to homeohydry, which is plants most are probably familiar with, that attempt to keep an internal water content to their liking, regardless of the water contents outside. I normally would say for the animal people in the room, probably this is mostly not animal people in the room, but you can think of this as somewhat analogous to ectothermy and endothermy in animals. So a corollary, though, of being desiccation tolerant, of poikilohydric and living on land is desiccation tolerance. There are lots of periods of time where there's no water potentially. And desiccation tolerance is just that plants can lose all free water and recover normal function upon rehydration. So if any of you have ever gone out for a walk after a rain, you've probably seen the mosses have greened up, and it's just amazing. But most mosses, it takes several minutes. So this one, they say 5 minutes. Some others could be 6 or 7. And if you try drying out and rehydrating a tropical moss, it could be 10 or 15 minutes. So they all can do this, but at different rates. Sometimes I get stuck in these videos and I don't know how to get out. All right. Sorry about that. All right, I made it. So a little bit more about desiccation tolerance. Plants that are desiccation tolerant can survive extremely low water potentials. And this trait or set of traits is rare in vascular plants, though it does occur, the desiccation tolerant ferns, for example. But it's pretty common in bryophytes. As you can imagine, bryophytes that live anywhere that it dries out will most likely be desiccation tolerant. This is the same species and potentially the same individual plant, hydrated on the left and desiccated on the right. So they really go through some extreme physical changes when they desiccate. Now, the genus Syntrichia has many dryland specialists, dryland including like semi-arid and arid landscapes. Many dryland specialists in this genus, and many of them play really important roles in biological soil crust, which is the soil surface in drylands, a community of cyanobacteria, other microorganisms that physically hold the soil together and fertilize the soil. Syntrichia is really diverse, though. There's a wide range of morphological and physiological variation. I've been working as a part of this NSF Dimensions of Biodiversity project to try and understand all these kind of components of diversity within this group. So there's a website there if you want to learn more about Syntrichia. And the most desiccation tolerant plants known are in this group, including Syntrichia caninervis, which we'll talk about today. So I showed you that other video, very cool, mosses are great, but this is Syntrichia caninervis. And in this, I sprayed some water on dry plants. And as you can see, it's very quickly opening back up and turning green before your eyes. This is completely real time, not sped up at all. So it's really fast. And we know from physiological studies that it can also be photosynthesizing this quickly too, depending on the conditions, but it's at least getting to work as soon as possible, recovery, whatever it needs to do. It's very good at this. Zoom does not like to, okay. There are genomic signatures of stress tolerance and desiccation tolerance in the genome of Syntrichia caninervis, including over 1,500 species-specific orphan genes, which are genes that don't have any clear homologs in other species. And many of those are associated with desiccation and stress response, high light tolerance as well. And there is a large expansion of this particular group of genes called ELIPs, which are known to be important in light tolerance and desiccation tolerance. So this trait, it goes deep into the genome. It's not trivial. But life as a desert moss has its implications, as I mentioned. Physiological ones include, of course, frequent and prolonged desiccation. And plants must tolerate excess sun even while dry, and they can't use any of it. There are accessory pigments that you're probably familiar with, known as carotenoids, that play various roles in helping tolerate excess light. They can absorb light for photosynthesis when hydrated. They can act as antioxidants, and they can function directly in dissipation of excess light, just releasing it as heat, which is protective. There are implications for growth and population dynamics. As you might imagine, with the means of fertilization being swimming sperm, they have a very slow growth rate. And this is kind of unfair to say. I feel bad saying it because I'm not sure that it's entirely the plant's fault. They have very little time in which they can grow. But even during that time, they don't think they're very fast growers. So an average growth rate, for example, for Syntrichia caninervis was 0.36 millimeters a year across 16 populations in the Mojave. So pretty tiny amount over a year. Sex ratio biases are common in desert mosses, and sexual reproduction is rare. In some species, Syntrichia caninervis, for example, there could be 3 times as many females as males, and some populations are entirely sterile and entirely genetically female. So they persist on asexual reproduction often. So actually, some of those findings I just reported were from my master's degree with Dr. Kirsten Fisher, looking at population genetics. But actually, while we were sampling for that study, came across this phenomenon of moss growing under these milky quartz rocks in the Mojave Desert. And I have to mention, I first presented this, the beginnings of this project at the California Botanical Society Graduate Student Symposium during my master's. So it's cool to be back now to give the sort of final report on the finished project. So my advisor at the time, Kirsten Fisher, now collaborator, flipped over a quartz rock and found moss growing underneath. And we had never heard of that or seen it before. So went to the literature and found just one report from 1989 about a subterranean moss greenhouse in the California desert. It was also in the Mojave. And they report a single patch of moss, Tortula inermis, growing under a piece of translucent crystalline limestone. And it really was just a report. It was a paragraph or something, just kind of noting the finding, but there was no other information about, what this habitat is like and where it occurs. So we were curious, kept digging into the literature, and found a large body of research on hypolithic communities that live under rocks, but mostly from bacterial community perspective. And some of these studies also noted in them that there were sometimes mosses in these hypolithic communities. So it's sort of known that it can happen, but very, very understudied. And actually, there's one species that I know of a rare obligate hypolithic moss that only lives under quartz rocks in Kansas. And so that's pretty neat. I would love to see that one day. And it occurs in exposed sites in grassland. So probably semi-arid and not a lot of shade. And we'll talk about what the rock might be providing for them there. So we set out to characterize this Mojave Desert hypolithic habitat. One goal was just to look at community composition, what's under there. As you can see from this photo, it looks different. This is all Syntrichia caninervis around where the rock was. And, from this distance, you can't tell what that is. It's green, and this is dark brown. That's about as much as we knew at first glance. To identify mosses, you could do some field ID, but the best is to take samples back and identify them in the lab. So we did that along some transects and 18 quartz rocks that had mosses under them for a total of 53 samples. Also wanted to know something about how much light gets through. So as you can see from this rock, it's not very translucent. And we measured the rocks that had moss, the thickness, and we calculated the light that can transmit, and also measured through 2 sample rocks, measured light ourselves, and wanted to characterize the microclimate, hypothesizing that it's different under there. And in fact, when we found this the first time, the mosses under the rock were still hydrated, and checking the weather, it hadn't rained in 2 weeks. So we had good reason to suspect that there's a different climate under those rocks. So we deployed temperature and humidity, relative humidity loggers for 5 months under rocks and at the nearby soil surface for comparison, and recorded every hour for 5 months. Like I said, to identify species, you have to do some microscopy generally, and especially because these plants looked a little different than their typical soil surface morphology. So we zoomed in on some characters like this leaf on or hair point, and cross sections of the leaves, and found very common in our samples was Syntrichia caninervis, the dominant moss of this site, so not surprising, and Tortula inermis. And this is what the plants look like all together. And there were also some samples that had Bryum argenteum, which is very common actually everywhere in the world. It's pretty much a weed. So that was also not surprising. We find that it's frequent in the desert, but not abundant, I would say. It's just a little bits everywhere. And this is sort of visualization of what the community looked like in the two microhabitat, hypolithic and soil surface. Syntrichia caninervis had a lot on the surface, less under the rocks, but still a fair amount. And Tortula inermis is the opposite to the extreme, almost entirely under in hypolithic spaces and only a few on the surface. And Bryum argenteum was, I guess, the most extreme, only one sample, and it was a surface sample and none in hypoliths. The thing that was interesting about this Tortula inermis finding is that that species is not very common at this site on the soil surface that we usually look at. It tends to occur lower elevations where it's hotter and drier in the desert. So given that we kind of had this hypothesis that the quartz rock would have a cooler, wetter microclimate, which we haven't tested yet, but it was surprising to see that this species would be restricted to the rocks almost at this site. So that is really what we aim to answer with the microclimate, is why might this plant be only under these rocks? So this is a graph of the temperature over the 5 months, and the line, the light blue is day and dark blue is night, and you can see these sort of day-night fluctuations. This is plotted the difference between the surface and the hypolithic. So above the zero, it's cooler under the rock, and below the zero, it's warmer under the rock. We also just calculated sort of the mean daily high and daily low, and as a summary. But I think you can see a lot from this graph that during the day, the rock tends to cool, and during the night, it warms the plant. And so they sort of experience this buffering from the extremes, because especially in this, later in the year, nights can be really quite cold in the desert. This is a higher elevation desert site. And so living under the hypolith can result in a buffered temperature. You can see here in December and then again in January, there are these periods where there's really no day-night fluctuation at all. And it stays consistently warmer slightly, but consistently warmer under the rock for several days. These are both snowfall events. So the area got blanketed in snow, and even then the rock provided a bit of warmth when it was probably quite cold. And then as spring, end of winter and spring, the pattern continues. So that was pretty enlightening, is that it wasn't just a cooler, wetter place. Well, it was cooler when it needed to be and warmer when it needed to be, perhaps. So what we could take away from this is that possibly Tortula inermis was actually benefiting not from this day cooler so much necessarily, but the warmth protection in the nights and in the winter. And Tortula inermis occurring at lower elevation, warmer temperatures may be a reflection of lower cold tolerance than the dominant Syntrichia caninervis. That helped make sense of that finding. And I think it makes a lot of sense personally. We also looked at the length of Syntrichia caninervis since it was abundant really in both habitats. I measured the stems, and the shoots that were under the rocks were 62% longer on average. And you can see this, just one sample here. This is a very typical looking surface plant, and this is what they looked like under the rocks. So one thought is that they just have longer growing periods. As I mentioned, they limited by when they can grow. And so that was the next question to answer with the relative humidity data and get an idea about the hydro periods and see if it is indeed longer under the rocks. Now, similar to the temperature graph, the 0, this is the difference on the y-axis, day and night are labeled, and over the 5-month period. And above the 0 is more humid under the rock, and below is less humid under the rock. And during the day when there's light and they need it, there's in this fall warmer period, at least, a bit more, I think 30% on average, higher relative humidity, daily high under the hypoliths. And at night, it gets actually a little less humid, but perhaps not when it's as necessary because there's no photosynthesis happening at night anyway. Then once winter, when the season changes, it stays pretty consistently more humid under the rock. And this is the growing season. So it seems quite plausible that they're experiencing much longer hydro periods and essentially longer growing periods. And that explains why Syntrichia caninervis is so much longer in these hypolithic habitats. Why this humidity difference may be here is it could be because of the temperature. It just doesn't allow it to dry out as much, or it could also have more condensation and actually providing more water. I'm not really sure, but regardless, it appears to provide buffering from desiccation, which happens very quickly in these habitats. The results for the light, we used this equation that was derived from geologists studying Mojave quartz, and using the average thickness of all of our rocks that bared mosses, we calculated 1.2% light is transmitted through the average rock. And just as a sort of gut check, I measured light myself through these 2 sample rocks using a total, this is 650 nanometer light, this 1.2%, but my measurements were sort of total light intensity, and the thicker rock transmitted less than 1%, and the thinner rock 4%. So the average seems right there within the range, and it's really a function of how thick the rock is. But also what was interesting about this paper that derived this equation is that there's this slight but perhaps important trend of this gray line is transmittance, how much is transmitted through, and smaller wavelengths are transmitted less than larger ones. And down here at this end is where ultraviolet radiation is. And so I became curious if it's not just lower light, but a different quality of light, different proportion of wavelengths may also be playing a role. So you may have noticed on the last time I showed this picture that the plant under the rock is very green compared to the soil surface one. And this brown coloration is pretty characteristic of this species. So the green ones, you do find variation in nature, but this was fairly extreme green coloration. It got me thinking about moss sunscreens, especially with that UV clue, or maybe it's just total light intensity. There are moss sunscreens mostly studied in Antarctic plants, and most are flavonoid compounds, most that have been studied. When you grow this species in the lab with no UV and very low light relative to the sun, they pretty much stay bright green. So there's something to either the light quality or the light quantity, which is what led me to the next experiment to try and figure that out. I wanted just to know about this coloration. Is it due to total light intensity or to UV radiation? So I went to the field. I went to Sweeney Granite Mountains Desert Research Center in the Mojave, in the Western Mojave. But before I explain what that was like, there's just a little bit of background that's important. I mentioned some of this, but light quality is important for photosynthesis. Blue and red light are real important, which you can tell from the absorption spectra here of the chlorophylls. They absorb in the blue and in the red mostly. And UV light, which is down here, is maybe damaging. It's typically thought of as damaging, but there's increasing evidence that UV radiation plays important regulatory roles, including widespread things like inducing defenses against pathogens, herbivores, and other stressors aside from UV itself. So it's important, even though we don't entirely understand why yet. And some of these accessory pigments do absorb into the UV spectrum. So there's a lot we're still figuring out. But also, as I mentioned, light quantity is important because they can only handle so much, and excess light will cause damage. So the carotenoids can play multiple roles. I actually mentioned this earlier, but here is a kind of proof of that. These are knockout plants that have the wild type up here is a normal plant, and these ones are lacking various carotenoids. And they measured over time and a high amount of light how much thermal quenching the plant does. And the normal plant does a whole bunch when there's a lot of light and then stops when it gets dark. But these ones that are lacking carotenoids don't do much at all. And so it shows the real important role of carotenoids in thermal quenching, which is, as a reminder, this like releasing extra light as heat as a protective measure. So my overarching question here was, how does Syntrichia caninervis tolerate intense UV radiation in the desert it lives in? And specifically, does it induce a sunscreen? And does UV radiation affect photosynthetic biology? The experiment consisted of I filtered UV radiation from these plants in the natural habitat, and then measured the effect on the color, photosynthetic pigments and antioxidants, and the transcriptomes to see what genes are involved. And I measured the recovery of photosynthetic efficiency from desiccation, just as a metric for how well they're performing photosynthesis. This is what the UV filtering setup looked like. This dark color here is all Syntrichia caninervis, and this is a UV filtering window. And for scale, this is 5 by 5 inches or so. And in a paired design, I set up an otherwise identical but UV transmitting window as a control for the window itself. At the same time, I collected site reference plants with no window at all, just to see the effects of the window itself. And there were 20 triplets like this, and I left them in place for 1 year. And at the end of the year, I collected the plants from all of the triplets. And first thing I noticed is that UV filtering causes plants to appear greener. This is just a sample of stems for demonstration purposes, but you can see when UV is filtered out, they are pretty bright green. And the normal sort of site reference, unmanipulated, are this very typical brown color. And actually the UV transmitted look sort of intermediate. Well, I did image analysis on a lot more stems and quantified the color, just proportion of green pixels out of red, green, and blue. And that really is what we're seeing here, is that UV filtered are the most green, UV transmitted in the middle, and the site reference, the lowest proportion green. So that was interesting and sort of confirmed to me that UV can cause this suntan effect. But if they're greener, the next question is, do they have more chlorophyll? So I measured chlorophylls, and actually, there's not a huge change in chlorophyll, but the chlorophyll effect that is here is there's actually slightly less chlorophyll in these greener plants. So there must be more of something else making the brown ones brown. There's also a higher proportion, a higher ratio of chlorophyll A to chlorophyll B, which is a high light response. In general, that's what plants tend to do when they're in high light habitat. So we took UV away and they respond as if it's sunnier. So this other color difference, then the next question is it due to other pigments? So measured a variety of accessory pigments and antioxidants, and several of them were not significantly altered by UV filtering, although this window effect is quite strong. But there were a few that were different, and in particular, zeaxanthin was higher when UV was filtered out compared to this control, and neoxanthin is lower when UV is filtered out. This is also a high light response for this zeaxanthin to go up and neoxanthin to go down. So again, a little weird. We took UV away for a year and the plants are responding as if they're in a sunnier place. So I looked to the transcriptomes for clues. I compared the number of gene transcripts in the UV filtered and UV transmitted samples, and there was a lot going on, of course, but some of the top most differentially abundant transcripts are involved in flavonoid biosynthesis. And they're higher, this transcript is higher when UV is filtered out. But looking into the function of these alphas xylosidase 1-like genes, they're involved in breakdown of flavonoids. So it seems that with with UV removed, there's an increased abundance of this gene transcript, which maybe increases breakdown of flavonoids, which should reduce total flavonoid content. Now, this is a hypothesis at this point because these genes haven't even been characterized for the precise function in this species, but it's intriguing to me. So this last part is that when you collect these plants from desiccation, you sort of need to give them some time. Although I bragged about how fast and good they are at recovery, they are compared to other plants, but the field adapted plants go through a lot. Lab plants are a bit more spoiled. So we put them in a growth chamber to allow them to recover just with water and measure their photosynthetic efficiency over this 192-hour period at 3 time points. And they stay hydrated from then on out, from time 0 to 192, they're hydrated. So at 0.5 hours, 30 minutes after hydration, measuring their photosynthetic efficiency, which is a quantum, it's like the electron transport part of photosynthesis, not the gas exchange part. the three groups, UV filtered, UV transmitted, and site reference, are not different from each other statistically and all very, very low. This is why we have to do the recovery, because people look at these, if you've ever worked with this type of data or seen it before, people will look at this and think this plant is dead or dying. But Syntrichia is cooler, just 24 hours later, and they're only in water in low light conditions. They have recovered a lot. Now, a healthy, sort of theoretically perfect plant is about 0.84. That's about as efficient as photosynthesis could possibly be. And so they're not there yet, but they are a lot closer. And interestingly, there's this staircase pattern that the UV filtered plants, have the lowest photosynthetic efficiency, and those that received UV have higher photosynthetic efficiency. This was very surprising, and at 192 hours, this pattern persisted. They didn't even quite make it to recovery yet, although maybe site reference did. And so this was very surprising that for some reason, removal of UV radiation from the natural habitat for a year inhibits recovery from desiccation and photosynthetic efficiency. So along with those high light responses we were seeing, what I think is happening here is that UV may be a cue for high light. And without it, the plant is unprepared for the light levels that it experiences, that it somehow uses the ultraviolet radiation as a metric for how much PAR to prepare for, or photosynthetically active radiation. So I looked back at the transcriptome to see if there were any other clues. And there are also 2 of the top 10 most differentially abundant transcripts were involved in stress response: polyamine oxidase and acyl lipid 9,3 desaturase-like. And in both of these, the transcripts associated with stress are higher when UV is filtered out. This is the very opposite of what I expected to find. We've all kind of experienced ultraviolet radiation on our own and know it's harmful in many ways, physical ways, although even in humans, it has important necessary health benefits. But still, I thought that if I took it away, the plants would do better. And they seem to be stressed, even though they're greener. So still working together, why? But what's clear is that UV removal in natural habitat increases stress for this plant, which plays out in recovery from desiccation and in photosynthesis itself. So to conclude, I'll just summarize everything we talked about. Your takeaways: Mojave Desert rocks provide a distinct microhabitat for mosses, including very low light with very little UV radiation and even a smaller proportion than it would get otherwise, buffering from temperature extremes and longer hydration periods. The hypolithic caninervis plants grow longer and greener than soil surface plants. UV induces a flavonoid, hypothesized flavonoid sunscreen, but definitely turns it brown. And natural levels of UV have beneficial effects on photosynthetic biology, and to the point that they will have evidence of stress if you remove it. And another thing that could be happening is that I mentioned that UV can induce a variety of protective mechanisms from pathogens and other stresses. So it may be that UV induces a response that protects from maybe desiccation. So those ellipses I talked about at the beginning are important for desiccation and for light tolerance. So these two tolerances are correlated, and this could be one of the links between them. And to just kind of put it all together, how do mosses survive and thrive in the desert? Well, I admit I've mostly talked about Syntrichia, but they are they're like the stars of the desert. They're very common, and many are restricted to deserts and drylands, very well adapted. So what they do anyway is desiccation tolerance. They wait out the rough times. UV tolerance, which makes use of all resources, even those that we might consider difficult resources to deal with. That's the adaptation part. Acclimation, they have high light tolerance, adjusting photosynthetic machinery as needed, as we've seen even when you change light for just a year. And they build up a suntan, which is presumably a UV tolerance or a light tolerance of some kind, although it has to be tested. And refugia can be important too. So definitely take advantage of more hospitable microclimates when possible. The quartz is a really excellent example of that because it's so extreme, but also just any granite boulder will do. It gives you some shade and causes a little bit more water retention. So these three things are really the key to how mosses survive and thrive in the desert. And they need really some of each to get both survival and thriving. Next steps for this project, I've sort of hinted at, but I would love to follow up on the identity and function of this presumed sunscreen pigment. I actually am currently exploring the evolutionary history of dryland adaptation in mosses and in Syntrichia using phylogenomics techniques. And I would love to compare patterns in Syntrichia, some of these things I found with other desiccation tolerant, darkly pigmented, but distantly related dryland bryophytes. Even liverworts sometimes have some of these characteristics. So it would be really cool to see if things are done the same way. Thank you all for joining me, and I have to thank my funding sources, many funding sources, including California Botanical Society, for funding this research over the years, many collaborators that have helped, and many student researchers that have been essential in getting a lot of this done. And of course, this was all done as part of grad school. So my community at UC Berkeley and Cal State LA is really important, as you all probably know. So the IB graduate students and support staff, the labs I was associated with, and my husband, who is my trusty field companion in these desert places. Thank you, and I'd be happy to take questions.